This is preliminary data on RNAseq of Aniridia iPS limbal stem cells reveal early defects in limbal development. So I'll just go through. We know Aniridia, it's caused by PAX6 mutations. We know it affects the eye in multiple ways. In the front, sorry, the laser again. It affects both the front of the eye and the back. But focusing on the cornea specifically, we know that it's quite a very strong limiting factor in these patients, that most of them tend to develop Aniridia-related keratopathy, characterized by limbal stem cell deficiency, cornea vascularization, and loss of transparency. So the cornea is composed of three main layers: the epithelium in the outermost part, the stroma, and the endothelium. And the epithelial layer is derived from the ectoderm, the surface ectoderm to be a bit more exact, and we know in these layers, in these tissues, PAX6 expression tends to be high and quite constant, while the stroma and the endothelium are derived from the mesenchyme and the neural crest, and in these tissues, PAX6 expression is lower and transiently expressed. And although it was always quite or widely thought and widely accepted that limbal stem cell deficiency affects primarily the epithelium, and PAX6 affects the deficiency affects the epithelium. It was recently shown that it's also necessary for neural crest migration and the formation, possibly the formation of the underneath layers. So the exact mechanisms and early events behind ARK are still unclear. So the goal of this project was to use IPS technology once again to model this disease and apply multi-omic analysis, particularly RNA sequencing, which detects and measures the global gene expression in the cells to uncover the disease mechanisms and understand limbal and corneal development. So once again, we differentiated iPSCs into limbal epithelial stem cells, and for this study we used two independent iPSCs from two independent Aniridia patients, both with nonsense mutations. And again, we used the same protocol as described before. At this point, we also tried to collect day 25 samples, although the Aniridia grew very poorly, so I'm still working on these at the moment. So anyway, we ran the wild type day 25, and this is an overview of all the samples analyzed. So IPS, or undifferentiated cells, we have both Aniridia and wild type here, day 15. I will go into detail on this in the next slide. Wild type day 25 samples, and then we added primary limbal epithelial cells as sort of a control. And what we see is that at day 25, the GO terms for the control are already linked to epithelial and epidermal development, epithelial development. But they're still quite far from, or a bit far from the primary, which suggests that they're still a bit immature. Anyway, because we only managed to analyze so far the Aniridia at day 15, these are the main results. We compared wild type day 15 in red here with Aniridia in blue. And what we see is that the GO terms enriched for Aniridia, so with high PC1, are linked to neuronal fate. And the wild type GO terms seem to be more mesodermal signature. We're still trying to interpret this. But what we also saw, and this was quite clear, was that the main pathways that seem to be different between two groups are extracellular matrix related pathways and TGF-beta signaling pathway. So looking into more detail, we see strongly reduction of several extracellular matrix related genes like proteoglycans. Here in blue are the downregulated, and in red is upregulated, and these are all wild types, and these are all Aniridia. We also see several collagens markedly underexpressed as well as metallopeptidases, which are important for the organization and the modeling of the extracellular matrix. And importantly, a lot of these genes are linked to corneal diseases as well. So for the TGF-beta signaling, we see the same trend. A lot of BMPs, TGF-beta 1, 2, and inhibitor downregulated in Aniridia versus wild type. And we know that TGF-beta signaling is important for corneal wound healing and epithelial cell migration. And then what we also saw, and something I'm working on, is several transcription factors are linked to neural crest migration, like PAX6 and PAX2 and more. So to summarize, we validated once again this model for Aniridia research. We show that RNAseq at day 15 suggests that these early defects might be more linked, I risk to say, to the actual limbal niche and the stroma than necessarily the epithelial. We didn't see any epithelial gene changes between the groups at this time point, but we are still validating these results. And the day 25 data does show an epithelial commitment in the wild type, and I'm looking forward for the Aniridia results, which hopefully we'll have in a couple of months. So thank you all. This was work that started at UCL, and now I'm continuing it at Radboud University in the Netherlands, and the collaborators as well at Tampere University. Thank you.
Thank you very much, Dulce. Any question from the audience? Yeah, please.
Please. You mentioned you had some isogenic cell lines. Were these ones the isogenics?
No, I didn't have. We don't have isogenic yet. We
So
want to. These are two Aniridia patients, separate, different mutations,
And the wild type?
and two independent wild type.
No
lines.
relatives to each other?
No.
Thank you.
Okay, thank you. One more question. We have 2 questions per paper.
You know that iPS, like embryonic stem cells, could behave as different state of naive versus prime. And so do you think could be a difference between the cell lines, the state of undifferentiated cells, that could explain the differences?
I don't think so. In this work, I don't think so. It could to a point, but I always look at the cells individually as well. And
Did you make an RNAseq on undifferentiated cells, at least to compare them at the beginning?
It's not in this data, but we have done it and we've added later. Yeah. But we have other iPS data here, so I added some. Not these iPS specifically, but we had some in the lab that our bioinformatician added. Ours are going in the next batch with the Aniridia, I hope. And they do cluster quite differently.
Okay. Thank you very much.
Thank you very much.
Thank you.